With this skill set becoming more commonplace, linked tourids become endangered in their home turf and branch out into new environments to avoid an attempt to overcome their population's common enemy. Thus, a new clade is cemented in history of the Fraditoroids, the symphony of linked tourids. Over time, with genetic mixing and interspecies competition, toroids continue to improve their body plans. Merging of the genetically similar linked and fibrillated toroids introduces a new colony of toroids. Injective toroids combine the competitive nature accustomed to the LTs with the flexibility and movement opportunities provided by the FTs, creating the most successful clade yet seen on this planet. They are the most efficient filter feeders so far. While the self-perpetuated genocide of linked toroids was taking place, the other factions have not been standing idly by. Researchers have deduced that this is due to their lack of legs. Instead, they have been growing their empires. There's only so much sulfides and hydroxides to go around. One viral infection was all it would take to change the course of the creatures of Symbiogena. By altering the genetic code of an isolated population of symbiogens, it forever transformed the self-replication protocol instilled into them over the generations. The virus created boil-like growths among the primordial sludge. Proteins that control the cancerous growth of the symbiogen body invert their relationship with signaling chemicals. Instead of promoting and protecting the growths, it caused a chain reaction to fill the tumor with fluid. The virus is even able to urgently kickstart the regeneration process and inject pluralistic gametogens into the tumor. Many a symbiogen died in this process from dehydration. However, the most resistant of the population were able to hold on to pass down the torch. Eventually, to combat this, the populations of symbiogen evolved to adopt a similar strategy by growing stalks above the primordial mat with sacs of exploding gametogens. How did this adaptation allow them to overcome the virus? It's because they were able to reproduce before their age weakened their immune system enough to allow the dormant virus access. Since the virus's DNA wiped out all but the fittest and strongest symbiogens, its DNA is forever entwined with the DNA of the remaining few. Now they are able to instantly recognize and resist the virus and closely related mutations of it. Populations that encountered this specific disease and pushed through gained the method of reproduction known as bursting. With their geographic isolation, such dramatic changes prevent it from mixing with virgin populations. Therefore, a new symphony of symbiogens emerges, known as Solicephia. The virgin populations, with their ignorance of the bloody battle long since waged, become the disconnected symbiogens. This symphony is titled Kommunukont. While we browse the land, we may turn our attention to the evolution of the members of Ectoderma. As the music crescendos, a cycle lapses, and a new symphony of beings emerge from the cacophony. Somatocysts are one of two symphonies emergent from the bifurcation of the Ectoderma mother realm. Low ionic strength solutions from freshwater located in isolated nearshore pits allowed for thick membrane assembly. Stirred by condensed gas outputs of land-based fumaroles, somatocysts found an unused niche in which to thrive. They are classified by their unique feature of nested body layers. The tissue that makes up the skin expands inward to provide functionality inside the body. By providing structural support, the gut of somatocysts advances to a stage that can handle larger prey than plankton. Side note, plankton refers to the microscopic organisms that cannot swim against the current. Creatures with this ability are classified as nekton. The structural gut structure of the clade may evolve independently in other unrelated groups. We call this cavity a coelom, from the Greek koilomat, meaning hollow or cavity. Successful as this advancement was, adaptive radiation quickly took over and bifurcated this group into two more subcategories. Orchestras at the somatocyst level have a major difference as to where they take this body plan. Groups in the southern hemisphere look to developing several organized structural gaps inside the body. This orchestra will be named vesicular somatocysts for its many vesicles, small fluid-filled sacs. Given free rein as to how to utilize these gaps, a plethora of varieties of early organisms emerged. Across the planet in the northern hemisphere, somatocysts expounded on the concept of the coelom by nesting multiple layers of gaps to the deep center of the body. This allowed for different chambers of digestion for different materials. This orchestra will be the lamellar somatocysts, for its many lamellae or thin plates found around the guts. What you are witnessing now is the asexual reproduction of a mature somatocyst. When this curious life form reaches the end stage of its life, it starts a 4-stage process to continue its genetic pedigree. Stage 0. Growth. For most of the somatocyst's life cycle, it spends its time growing, processing nutrients, and surviving the seasonal changes of the freshwater pools and lakes. Near maturity, the body starts producing chemical signals that alert it to begin the reproduction process. The signal can be kickstarted by a variety of conditions such as the concentration of DNA damage, severely stressful conditions, starvation, cold temperature, and a low local population. Stage 1. Synthesis. The somatocyst diverts all available energy to the formation of precursors necessary to bear children. Body parts, such as redundant tissues and inactive lysosomes and ribosomes, are recycled. At this stage, gene-carrying protein chains organize at the two axes along the body and duplicate. The rapid rate of duplication causes increased chances for mutations. The higher the stress level for the creature, the faster it must reproduce, and thus the likelihood of harmful mutations can be high. Stage 2. Recomposition. During recomposition, the cyst shrinks down to 1/20th of its original diameter. This allows for the disposal of even basic necessities for life. The cytoskeleton is dissolved. Now, like a caterpillar's cocoon, much of the somatocyst is reduced to a biochemical gel with instructions to form immature seedlings. Stage 3. Expansion. In the expansion stage, the somatocyst reforms itself into a form unrecognizable from its original. Pods are produced along the central spine and balloon outward. Byproducts of metabolism that are normally excreted are instead shifted toward the polar ends of the cyst. This keeps them away from the defenseless gametocysts with no immune system at all. Stage 4. Burst. Once the pressure of the gaseous and liquid byproducts reaches a critical level, the cyst bursts. Each daughter is left with a packet of essential nutrients to last the first week of survival. Oftentimes, not all babies form correctly and leave behind a tightly packed husk of organic material. Known as a dud kernel, these serve as a nutritious food source for other land-based organisms, such as primati. Diametric to the somatocysts, the cymatocysts engage in a natural yet unfamiliar mode of life. By manipulating and responding to the flow of cellular migration, they are able to take on unique body plans and form complex relationships between organs and tissues which comprise them. Hold on though, let's start from a more familiar place. Members of Ectoderma mother realm utilize pockets of naturally occurring fatty membranes to incorporate their bodies. In time, cymatocysts develop organ systems depending upon these liposomic balls that they start to develop inside of their tissues. Through differentiation, the lipoids start taking on other functions in the cell. Factories in this body tend toward two islands of stable behavior, one to produce turbulence in the cell structure and one to divert the flow into usable patterns. Over time, the factories differentiated to produce only the type assigned to them. The factory first relinquishes a thread of bilayered phospholipids, the phosphorus being a gift of the island's fumaroles. The threads merge inside a structure designed to cluster them into shape. This ball of fatty threads is called a complex. Small complexes surrounding an older, larger complex serve as secondary complexes. Complexes are created with adhesion proteins lodged in a ring around the membrane along one axis. This allows them to form capsules to contain other structures. Stay with me now. The other type of factory produces harder spheres with imperfections on the surface. The name of the structure is a dimploid or noid, and it somewhat resembles a golf ball. There are four main categories of dimploids in a typical cymatocyst: pitted dimploids, hexagoids, canaloids, and resinoids. Pitted dimploids are isolated structures often surrounded by a perimeter of specialized secondary complexes. As they rotate, they are able to transfer vital nutrients and waste from one complex to another. You can think of the dimploid as a mailing center and the complexes as a local neighborhood. Hexagoids are dimploids whose shape does not allow for easy input and output as freely as the pitted dimploid. By using a hexagonal honeycomb structure, these can grow to any necessary diameter as the pattern is infinitely tileable. In the deeper recesses of the structure's walls, nutrients and other molecules can be stored for long periods of time. By contrast, canaloids serve a more active purpose. With channels etched around the circumference of the structure, carved in a single direction, these dimploids can rapidly transfer large quantities of material. However, a signal can be sent to one or more canaloids to rotate perpendicular to the free flow state. This effectively stops material transfer in that section of the body, and nutrients are routed back to other areas. A neat side effect of this method of transportation is that the Cymatocysts can trap unwanted foreign bodies by isolating material in a canaloid tube and either expel the unwanted guest or eradicate it. Finally, there are dimploids to produce the turbulence that govern intercellular migration, known as tympanic dimploids or resinoids. These structures are classic dimploids covered by a thin sheath on the outside. By taking a cross-section, one can see the inner workings. Beneath each dimple along the surface under the sheath, there is a small cavity filled with vacuum. Attached to the bottom of each cavity are fibrils that connect it to the piezoelectric proteins dispersed throughout the structure. When activated, the proteins pull on the cavity, retracting the sheath into the dimple. Once the signal has worn off, the sheath rapidly accelerates back to its natural smooth disposition. Repeating this cycle hundreds or thousands of times per second creates a pressure wave inside the cyst that manages cellular migration. The signals produced by one resinoid are filtered through layers of complexes and interact with the waves of other resinoids, creating a non-local, decentralized network of signals interweaving the body. Since each resinoid vibrates weakly at a high frequency, they must work in sync to generate low Hertz beat frequencies. Movement of cells caused by resinoids can be described with acoustic streaming, which is based on the Navier-Stokes equations. Resinoids can also have various effects on the surrounding complexes by modifying their structures to resemble one of the following shapes: liquid, crystal, sponge, crystallite, fiber, micelle, oligomer, or monomer. Each comes with its own use cases and properties. Properties that govern the shape of the whole Cymatocyst habital are as follows: resinoid network, pliability, hardness, buoyancy, mass, and shape. Pryists that incorporate any sort of silica into their body structure belong to the Lapis symphony. They take silicon dioxide from their environment and encapsulate themselves with a hard frustule like diatoms from Earth. As helpful as this is for their survival, it also serves as a key component to future organisms that would like to harvest a concentrated material. Lapis organisms are among the most beautiful on the planet as they exemplify the power of organic life to harness mathematics and the infinite possibilities of organization hiding in latent space. Many planktonic pryists drift around the tops of the oceans. Danger awaits them daily from the UV radiation of the sun threatening to dismantle their very genomes. However, the laser in the sky can only pierce the water so deep, leading to a successful motif of pryists named Melanoplasts. Identifying the presence of light is a task not easily accomplished. In Melanoplasts, pockets of nerves near pigmented bits of membrane react to the presence of electromagnetic radiation. By paralyzing the mobile amoeba-like extensions, this causes the creatures to sink underneath the waves. With less exposure to damage, Melanoplasts carved a valuable niche for themselves. Over time, the pigment covers most if not all of its body. The pockets of pigmentation are also useful for redirecting light used for food toward the energy centers from any angle. To accomplish this, the habital is transparent under the black layer. Following a separate but related line of methodology, Chromatoplastids learn to harvest the energy from the sun in a more efficient way. The success of primary producers is overwhelming, and they diversify into organisms sporting nearly every color of visible light. They avoid rupture by blocking out ultraviolet radiation from the sun. Another creature returns from the previous episode in the Interoplastids. These are still single-cellular like the Chromatoplastids and Melanoplastids. I included them in this episode so that you can see where they exist within the phylogenetic tree. To reiterate, Interoplastids are transparent fatty cells that devote much volume of their bodies to storage of fats, proteins, and polysaccharides. To wrap up the plastids, we can find Viridioplasts, waving from the floor of shallow salty waters. They soak up red, yellow, and orange, and some blue light, reflecting a bluish shade of green. As the producers of their local ecosystem, these habitals will have a large impact on the direction of the first herbivores once they arrive. By tangling their single-celled cilia in knots, these habitals learn to act as one in a group. In reproduction, gametoplasts are released into the water with a fluid adhesive substance. This causes them to thrash about and stick to other gametoplasts. When a tied-up group breaks free of the jungle of knotted fiber, they float away in order to start life anew on a sturdy piece of rock or sediment. Solicephus is a very flexible branch of life. While plastids are stuck taking on a twisted journey across time, Solicephus experiments with spreading its seeds in novel ways. Bursting reproductive stalks made way for a safer method, where the stalks atrophy quickly after emerging. They dissolve into thin strands that hold on surprisingly well to the distal sacs, eventually caving under the tension and releasing the unburst sac through the currents. Even further along in time, the remnants of the stalk attached to the sac form a structure to help the gametogens get away from the mother in order to not compete for resources. This deviation from the mean became so distinct from its original behavior as to produce a new lineage named Buddosymbiota. The creatures of this clade experiment with sacs that are hard and brittle, and sacs that are spongy and flexible. It turns out that flexible sacs succumb easily to cold temperatures, and brittle sacs are too easily shattered by environmental hazards. Thus, brittle buds exist closer to the poles, and flex buds live in tropical zones closer to the equator and disperse with the global currents. Underneath the shallow seas of the wet expanse, Cymatocysts harness the properties of canaloids to their extreme. The waters are as of yet still too acidic for calcium carbonate materials to take shape. Any attempt to generate the hard compound will see it rapidly dissolve before becoming useful. Thankfully, the massive number of priests floating in the waters can help overcome this issue. Seasonally, priests will undergo a massive die-off, where the sky is overcast for months at a time, and their diatomaceous bodies sink to the ocean floor. Cymatocysts can use specialized enzymes to break down and extract the silica from their skeletons. Through a biomineralization process, these organisms can interpolate their matrix with a durable compound to produce a kind of hard shell or skin. By organizing canaloids into a chain, Tubiformes form a long tube covered by psylloid, which is composed of particles of silicon and polysaccharides and sodium hydroxide. In addition, the organisms also develop new organ system out of repurposed canaloids. Instead of a hollow tract in the canal of the dimploid, the organism creates rotating teeth that help move the particles along. With a slight modification, these teeth can act as gears to interlock. This adaptation would be seemingly useless if it wasn't for the simultaneous change of a structure reminiscent of a tail coming out of one side. When perfectly aligned, these gears can link up and become capable of movement faster than cellular manipulation. In fact, with many gears in a row, this movement becomes a snap so fast that not even the human eye can follow. This ability of a cymatic twitch lays the groundwork for true greatness in its descendants. Ligitoroid is a classification of toroids that design their body plans based on the properties of twisted fiber. When you take a piece of string and fold it back on itself, then give it some twists, you can turn a flimsy fiber into something like a rope. Its capability of tensile strength drastically increases while decreasing the amount that it can stretch. When pulled, a component of the force that would be acting along the length of the fiber is redirected perpendicularly to the string itself. Their bodies become labyrinthian as they undergo complex knotting behavior, usually connecting back on itself like a Mobius macrotoroid, with the twists of a helical toroid.